Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, so picture this. What if, the deadliest enemy you could imagine had a secret weakness, like a hidden self-destruct button, just waiting for us to figure out how to turn it back on?
Interesting.
And what if I told you that for some really aggressive cancers, this might actually not be science fiction anymore?
Really?
Yeah, we're going deep today into some brand new research that suggests that scientists may have found a way to reactivate this self-destruct mechanism inside these cancer cells.
Wow, that's pretty amazing. So, what's the science behind this?
Well, it seems cancer cells are pretty sneaky. They figured out how to silence their own self-destruct signals. They're basically manipulating their internal systems to make sure they survive and keep growing. But researchers are now figuring out how to flip those switches back on. This could be a total game changer for how we fight these really tough cancers. So to really dig into this, we're looking at two sources today. There's a really in-depth scientific paper from Nature Communications and also a super interesting health news article from medicalexpress.com. They both came out just this month, March 2025, and they both highlight this new potential treatment strategy. And our goal today is to break down this really exciting research and try to understand what it means for treating these really aggressive cancers without getting lost in all the super technical jargon.
Yeah, I think that's really important. Making this kind of research accessible is key.
Absolutely. So at the core of this research is how cancer cells are basically manipulating their own internal instructions to survive and grow unchecked. It's kind of like, think of it like this. Our genes hold the complete instruction manual for how our bodies work, right?
Like a blueprint.
Exactly. But the way these instructions are actually used isn't always straightforward. There's this critical step called alternative ribonucleic acid splicing.
Right. Alternative ribonucleic acid splicing. That's a mouthful.
It is. So maybe you can help us understand what exactly happens during alternative ribonucleic acid splicing.
Okay, so imagine you have a single recipe for, let's say, a simple vanilla cake. Pretty basic, right?
Right.
But what if you could tweak that recipe? Like add some chocolate, maybe a bit of coffee, change the baking time a bit, and end up with a bunch of different cakes, all starting from the same basic recipe.
Oh, I see.
Well, that's kind of what happens with alternative ribonucleic acid splicing. It's like the cell is a master chef taking a single gene's instructions and creating a whole variety of different proteins from it.
So it's about making the most of one set of instructions to create a lot of different outcomes.
Exactly. And this ability to create a whole bunch of different proteins from one gene is what makes our cells so adaptable. They can fine-tune their activities based on what they need to do at any given time. It's really remarkable.
So how do these cancer cells exploit this whole intricate editing process for their own advantage?
Well, think of it like this. Cancer cells are master manipulators. They basically hijack this splicing process, like they're taking over the editing room. They're rewriting the script to make sure the story ends with them winning, even if it means harming the rest of the body.
So cancer's like a rogue editor, cutting and pasting genes to create a version of the story where they always come out on top.
Exactly. And one of the key players in this whole manipulation is something called poison exons.
Poison exons. Those sound a bit ominous.
Yeah, the name is kind of fitting. Think of them like built-in kill switches for protein production. When a poison exon gets included in a ribonucleic acid message during splicing, it's like a big red flag telling the cell to destroy that message before it can be used to make a protein.
So it's like a self-destruct sequence for the genetic message.
Exactly. So in healthy cells, these poison exons are super important for regulating protein levels. They prevent the cell from making too much of any one protein. It's like a safety mechanism.
Right, so they're like natural regulators making sure things don't get out of control. And in cancer cells, what happens? Does this regulation break down?
You got it. In many cancer cells, this whole poison exon system gets disrupted. The off switch doesn't work properly.
And this brings us to a specific gene, right? This TRA2 gene that was a key focus in this study.
Exactly. Scientists at the Jackson Laboratory in UConn Health zeroed in on this TRA2 gene. They found that cancer cells are actively suppressing the activity of a poison exon within this gene. They're essentially turning off the off switch.
So they're deliberately making sure the kill switch isn't working. But why? How does that help the cancer?
Well, by suppressing that poison exon, cancer cells can churn out massive amounts of this TRA2 protein, and too much of this protein acts like rocket fuel for cancer growth. It's like they've found a way to supercharge their own development, helping them grow uncontrollably and even evade the body's natural defenses.
That's unsettlingly brilliant. So did the researchers find any link between this suppression of the poison exon and how the cancer actually behaves in patients?
They did. It's a pretty strong and worrying correlation. They saw that when poison exon inclusion in the TRA2 gene was low, meaning that off switch wasn't being used very much, patients with a whole range of different cancers had worse outcomes.
So this isn't just limited to one specific type of cancer. It seems like a broader issue.
Yes, and it was especially noticeable in aggressive cancers like triple-negative breast cancer and certain brain tumors, but they also found it in ovarian cancer, skin cancer, leukemia, and even colorectal cancers. It's a pretty widespread vulnerability.
Wow, so this is a really big deal.
Yeah, this correlation across so many different cancers really points to how important this TRA2 poison exon mechanism is in how cancer develops. And this discovery is what led the researchers to start investigating a potential therapeutic strategy, a way to, flip that switch back on.
And that strategy involves something called antisense oligonucleotides. That's a bit of a mouthful.
It is, isn't it? Antisense oligonucleotides, or ASOs for short.
ASOs. So what are they in simple terms?
Well, imagine them as tiny, highly specialized tools, almost like microscopic wrenches designed to tweak specific parts of the cellular machinery. They're actually synthetic ribonucleic acid fragments carefully engineered to bind to very specific ribonucleic acid molecules inside a cell.
Okay, so they're like targeted ribonucleic acid fragments. What makes them so special?
Scientists can design these ASOs to have a unique sequence, like a code, that allows them to target a very specific ribonucleic acid molecule. In this case, the one produced by that TRA2 gene we've been talking about.
Right, that gene the cancer is manipulating. Yep. So how do these ASOs help bring those poison exons back into play?
It's really clever. These ASOs can be designed to basically force the cell to include those poison exons in the TRA2 gene's ribonucleic acid message. They interfere with the cancer's ability to suppress that off switch.
So they're like little molecular saboteurs, sneaking in and disrupting the cancer's plans.
Exactly. And when the poison exon gets included, well, you remember what happens then, right?
The whole ribonucleic acid message gets destroyed, right?
Yeah.
Which means less of the TRA2 protein is produced.
Bingo. By getting that poison exon back in the game, those ASOs help the cell regain its natural ability to break down all that excess TRA2 ribonucleic acid. And less ribonucleic acid means less protein, which ultimately slows down or even stops the tumor's growth. It's like hitting the brakes on the cancer's runaway train.
That's incredible. And I remember there was a quote from one of the researchers in the article, something about a rheostat.
Yes. Nathan LeClaire, one of the lead researchers on the study, he explained that these ASOs are like a rheostat, a dimmer switch for protein levels. They can fine-tune the amount of protein being made rather than just trying to shut it down completely.
So it's a much more controlled approach than just trying to wipe out a protein altogether.
Precisely. And there was another really interesting finding. The researchers also tried completely removing the TRA2 protein using CRISPR gene editing. But even when they did that, the tumors continued to grow.
Oh, wow. So even getting rid of the protein completely wasn't enough.
It seems that way. It really suggests that targeting the ribonucleic acid with these poison exons might be a much more effective strategy than just focusing on the protein itself.
That's surprising. Why would targeting the ribonucleic acid be more effective than getting rid of the protein completely?
That's the million-dollar question. Olga Unsuko, another lead researcher on the study, offered a fascinating hypothesis. She suggested that this ribonucleic acid, the one with the poison exon, might be doing more than just blocking the production of the 2A2 protein. It might also be trapping other important molecules within the cell's nucleus, creating a very unfavorable environment for the cancer cells.
So it's like a double whammy. Less of the bad protein plus this toxic ribonucleic acid wreaking havoc.
Right, so it might not just be about the amount of the 2A2 protein, but also the presence of this modified ribonucleic acid that's really messing with the cancer cells.
This is all so fascinating. So how does this research impact the bigger picture of cancer treatment?
What has the potential to revolutionize how we treat these very aggressive cancers, especially those where current treatments just aren't very effective. And one of the most promising things about these ASOs is their precision.
Right, the article mentioned that they seem to be very targeted in their action.
Exactly. early data suggests that these ASOs are really good at zeroing in on the cancer cells without causing much disruption to healthy cells. And that's huge because it means we could potentially avoid those nasty side effects that often come with traditional cancer treatments like chemotherapy.
And could this approach of using poison exons be applied to other cancer-driving factors as well?
That's the hope. Researchers believe that this strategy of targeting poison exons could work for other ribonucleic acid-binding proteins that contribute to cancer, not just Ta2. It could open up a whole new world of highly targeted cancer therapies.
It all sounds so promising. But of course, we need to be realistic. It's still early days for this research. What are some of the challenges or limitations they still need to overcome?
You're absolutely right. More research is definitely needed. Scientists need to refine these ASO-based therapies and figure out the best ways to deliver them directly to tumors inside a living organism. It's one thing to make these things work in a lab, but getting them to work effectively inside the body is a whole other challenge.
Right, because our bodies are complex systems. It's not as simple as just injecting something and hoping it hits the right target.
Exactly. The next big step will be to test these ASOs in animal models to make sure they're safe and effective, and to figure out the optimal way to deliver them before we even think about human trials.
And what about the possibility of unintended effects? Even though early results suggest these ASOs are very specific, is there still a need to investigate that further?
Oh, absolutely. While the initial lab results on specificity are encouraging, we need to be thorough. Figuring out whether these ASOs might have any unintended effects on other genes or cellular processes, what we call off-target effects, is complex and will require a lot more research in more complex biological systems.
It's important to be cautious and thorough when it comes to something as crucial as cancer treatment. So to sum up, what were some of the key takeaways from the researchers themselves? What did they emphasize?
Olga and Zuka really highlighted the strong correlation they found between those low levels of poison exon inclusion in the TRE2 gene and the poor outcomes across a whole variety of aggressive cancers. That finding has really significant implications for how we approach treatment.
And Nathan Leclerc emphasized that rheostat analogy, the potential for these ASOs to precisely adjust protein levels in cancer cells, making them a potentially very effective and targeted therapy.
Exactly. And Onzuko also pointed out that targeting the ribonucleic acid and promoting that poison exon inclusion might be doing more than just reducing TRE2 protein levels. It could be disrupting other ribonucleic acid binding proteins that are crucial for cancer cells to survive.
So it might be a multi-pronged attack on the cancer, not just taking out one protein.
Precisely.
This has been a really fascinating look at some groundbreaking research, and it raises an intriguing question. If we can manipulate ribonucleic acid splicing in this way, what other cellular mechanisms that go wrong in diseases could we target with similar strategies?
That's the big question now. It opens up a whole world of possibilities for new therapies. It'll be fascinating to see how this field develops and what new targets and treatment strategies emerge as we continue to unravel the complexities of our cells. It's a really exciting time for medical research.
Absolutely. Thank you for joining us for this deep dive. Until next time.
Thanks for having me. It was a pleasure.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.